Hello, this is Dr. Richard Fleming, and today is Monday, August 31st, 2020. I've been getting a lot of feedback from individuals telling me that they have been researching different COVID 19 issues, including the numbers of cases, the cause of deaths, terrain, cult versus an actual scientific germ theory. And want to take this opportunity to point out the difference between science and journalism. Journalism is typically involved in researching things by reading what other people have said, and then you go talk to those people and you report what they have to say and you compare things that other people have written. That's not science. Science begins with learning what has been done in a field before. And that can either be through classroom training or by working with colleagues or professors, or typically when you begin to enter the field of science, you get sent to look up a topic. Now, when I started back in the Stone Ages, we actually went to libraries where we went to the card catalog and looked up topics and then went through mounds of journals on different floors of the library to try to find different journals, read the papers, look at the references in those papers, go try to find those papers to get some idea of what the actual topic field consisted of, the prior work that had been done, determine what was credible, what wasn't credible. Today, that really kind of amounts to, for most people, going to Google. And that typically means that for most people, what they consider research to be is actually uh, this. Sorry about that. Actually, this. This is not research, and this person is not a well, maybe this type of researcher, but not a legitimate researcher. Research is the process of getting that information and then going back and making some conclusions about what information may or may not already exist and trying to formulate some type of idea about what you would like to investigate. And that means taking all that prior information and then trying to develop an experimental design, the methods as it's called in science. That is, what types of patients, or if you're not working with people, what type of cell cultures or chemicals or laboratory benchmark lab materials you need to then introduce a variety of variables, either treatments or chemical reactions or vaccines or genetic coding sequences, something that you're going to do eh, to measure an impact of something else that you do. So, for example, with COVID 19, with the NCT 04349410 protocol that I developed, it's a matter of looking at people who have COVID 19 by PCR swab testing. And you can either take somebody who's symptomatic in the hospital or symptomatic pre hospital and then choose a number of treatment arms. These are the different treatments like hydroxychloroquine or remdesivir or azithromycin or interferon or interleukin 6 inhibitors or any of a number of other treatments, and then to use those treatments with those patients and then measure an outcome. And unfortunately, many of the studies that have been done so poorly that the end result is either the patient died or didn't die, or how soon did they get out of the hospital, and poorly done, not even differentiating differences between people being on ventilators or not. So in the study that I've set up for national clinical trials, we actually measure not only blood tests, which look at the virus itself, but what the body is doing chemically in response to the virus. So that might be things like interleukin 6 or fibrinogen or a number of other responses. But then we also couple that with an absolute quantitative measure of the actual reaction to the virus in the lungs itself. That's FMTVDM. And then we measure that over a period of time and see what effect a treatment actually has. Research is not going on Google. Googling things and getting background materials and asking what somebody else has done forms the very first part of the paper, which is called the background material. And a real research paper begins with background material, but then builds on from there with the methods and how the study was done and how you're going to look for changes and how you're going to statistically analyze the results to determine if those changes were just due to chance or if they're actually due to some type of consequence of the treatment. And then you want to discuss that information after you present the results, and you can even wrap it up with a little conclusion. Sometimes you can put in limitations or not, which, quite honestly, for anybody who's in the field, those limitations are obvious, but then you would propose what the next step in the research should be. That's research. Googling things is not, going to the library is not, talking to somebody on the telephone is not. Research is real scientific results of taking what's known, applying a method and a scientific method to measure outcomes as a result of something you do in the individual or whatever experimental design it is, analyzing whether those results are significant, and then reporting how that fits into the general scheme of things to give a better understanding. In other words, it's everything that really hasn't been done in the papers that you've been seeing up until now. And for those of you who consistently tell me you've been researching COVID 19, what you've been doing is reading what other people have been talking about with COVID 19 or terrain cult or whatever it is that you've been doing. That's not research. It doesn't make you a researcher. And while I appreciate the fact that you're doing it, it doesn't bring any answers to what's really going on.